Chapter 8, Tools of Recombinant DNA Technology. The learning objectives for this topic are: describe how gene researchers use mutagens, explain the function of and use of reverse transcriptase in synthesizing cDNA, explain how gene researchers synthesize nucleic acids, describe 3 uses of synthetic nucleic acids, explain the source and names of restriction enzymes, describe the importance and action of restriction enzymes, define the vector as the term applies to genetic manipulation, and explain significance of gene libraries. So the subtopics for this topic are: we're going to talk a little bit about history, we're going to go over mutagens again with an eye toward using them in biotechnology, use of reverse transcriptase to synthesize cDNA, synthetic nucleic acids, restriction enzymes, vectors, and gene libraries. As promised, here's the short history of the tools of recombinant DNA technology. So until recently, we were limited to working with naturally occurring organisms and their mutants. Well, in the 1990s, we developed tools to insert genes pretty much wherever we wanted. I put that in quotes because it's not as easy as it sounds. And it's faster than the traditional process, and it's also more accurate. So let's talk about mutagens and reverse transcriptase. We went over mutagens in the last chapter, but let's take a look at them with an eye toward how we use mutagens within recombinant DNA technology. We use both chemical and physical mutagens, and this creates mutants faster than usual. Probably not so good for the mutants themselves, but since we can identify the ones that we want, it speeds up the process. So here's an example. The penicillium that we currently use for making penicillin produces 25 times the penicillin that the Fleming strain or the wild type strain has. So here's a situation where we were able to use mutagens to create an organism or to breed an organism that produced the product that we wanted. Reverse transcriptase. Reverse transcriptase is an enzyme that is produced by retroviruses. HIV is a prime example. HIV is a horrible disease, but reverse transcriptase has been a valuable tool. What it does is it makes DNA from RNA. That's why we call it reverse transcriptase, because with the central dogma, we always go from DNA to RNA, and we don't go back the other direction. Well, with reverse transcriptase, you can. Now you're probably wondering, why would we want to do that? Well, we have hundreds of mRNA copies of each active gene, where we only have one copy of the DNA gene. So it's a whole lot easier to isolate the mRNA, use reverse transcriptase to make DNA from it. We call it cDNA because it's a copy from mRNAs. And then we can insert that into an organism. So we can take eukaryotic genes and put them into prokaryotic genes. That's where we take the modified mRNA, not the one in the nucleus, but the one that's in the cytoplasm. All the introns are removed. We make a DNA copy, put it into a prokaryotic cell that is not set up to remove introns. Isn't that cool? As an example of how we use this one, that's how we got human insulin, through the reverse use of reverse transcriptase to make a copy of cDNA from a mature mRNA. On to synthetic nucleic acids. First off, we can make nucleic acids in the lab, and we call this cell-free synthesis. Now the reason why we can do this is enzymes will work whether they're in a cell or whether they're in a test tube, as long as they have substrate and you give them optimal conditions such as temperature, pH, etc. So in this way we discovered the codon code, and we were able to use the codon code to come up with more synthetic DNAs. So we can create genes for specific products. Once we know what the amino acid sequence is, then we can go in and make DNA for it. Human insulin is my example again. We could go through and keep making cDNAs from mRNAs, but once we know what the code is, once we've gotten the cDNA, we can make copies of that cDNA using synthetic nucleic acid processes to keep pumping out the copy that we already have in a cell-free environment. Kind of cool, huh? We can make DNA and RNA probes. Now we'll be talking more about the uses of these when we talk about applications of recombinant DNA techniques. And we can synthesize antisense nucleic acid molecules. This is becoming a big movement of antibiotic researchers. It's non-antibiotic, and hopefully it will help us fight organisms that have become resistant to all of these safe antibiotics that we're using. Restriction enzymes are pretty much DNases. They are enzymes that cut DNA, and they are bacterial enzymes that help protect them from phages. So foreign DNA comes in, they use restriction enzymes to cut them up, hopefully before they cut up the bacterial chromosome. It's a case of who's quicker than whom. So bacteria recognize their own DNA and protect it by methylating their own DNA, whereas phages usually come in unmethylated. Restriction enzymes cut at restriction sites, and they vary from bacteria to bacteria. And they are often palindromic sites, so you remember palindromes from transposons. These are stretches that are the reverse of each other. So let's talk about how restriction enzymes actually work and how they're used. So we have a restriction site, it's a palindrome, and the restriction enzyme comes and clips here. Now in this example we have sticky ends, where we have single-stranded DNA that are hanging out. So if we use the same restriction enzyme on two different organisms, then we can do a switcheroo and create recombinant DNA molecules. And we always add a ligase so that we can heal the nicks. These are easy to make recombinants. These sticky ends, they want a hydrogen bond with their matching partners on the other side of the DNA strand, so they're fairly easy to make. But you can only use this when you can find the same restriction site in the gene that you're interested in the organism that you're interested in. So sometimes we go with restriction enzymes that make blunt ends. They just snip straight through. So we can use two different restriction enzymes that make blunt ends, and we don't have to worry about these sticky ends matching up. Then we use a ligase to do the recombinant DNA molecules. Now this is harder to do, but we can insert where other restriction enzymes have made cuts, so we have a broader range of what we can do. Now examples of an restriction enzyme that makes sticky ends would be Echo R1. So how do we get the name? This is for E. coli strain R, and it's the first one that we found in strain R. Now for blunt-ended restriction enzymes, or restriction enzymes that make blunt ends, this one came from Sericium marcescens, and it's the first one that we found, and this one came from Haemophilus parainfluenzae 1. So that's how we name them. On to vectors. Now you may have thought of vectors as, say, houseflies or mosquitoes, insects that carry disease. Well, within the realms of recombinant DNA technology, vectors are anything that can be small enough to be manipulated in the lab that can carry genes into the new organism. And they have to be able to survive in the cells. Sometimes you can put DNA in, and those restriction enzymes, if you're putting it into a bacterium, go snip, snip, snip before you get a chance to recombine with the chromosome. So they have to survive in the cells. So let's take a look at some recombinant DNA technology vectors. First one are plasmids. Plasmids are the most stable. They're circular, bacteria are used to seeing them, and they're not going to get snipped up by restriction enzymes. So we have a tendency to use plasmids a lot. Besides, they're easy to manipulate. Transposons and phages, they're not quite as easy to use. They have to be able to survive within the cell and be inserted into the chromosome before they're degraded. There are ways of doing that. Now, once you get the gene that you want onto your vector, you have to put a tag on it. The tags show you that you have been successful in inserting the gene of interest into your organism, that it has successfully recombined. and we have a number of different tags. One of them is the enzyme for metabolizing a specific nutrient. So if you have, say, E. coli that is unable to metabolize, say, cellulose, and we put in cellulase right next to the gene that we're studying, what you do is you take your organism, grow it on cellulose-containing media, whatever survives, you know it's recombined. Antibiotic resistance can be used the same way. We have a tendency to use that one more frequently. And we also like GFP, green fluorescent protein. Now, back in 201, you made use of antibiotic resistance to show that your little bacteria had been transformed, and then we used the green fluorescent protein to show the expression of the gene that we were actually looking for. Now, with the experiment in 201, that was pretty much just for fun, to show you that it could be done and to talk about transcription, translation, genes being turned on, genes being turned off, but they're also used in real life in real labs. So let's take a look at how we would make a vector. So we would take a plasmid that is our vector, and it's got an antibiotic resistant gene in it. This is going to be our tag. Here's our restriction site, and here is what we want to put into it. So in this case, we're looking at human growth hormone, HGH. You may have heard an awful lot about this one. There's popular diets that use it. So we take our mRNA, which was easy to find, use reverse transcriptase on it, and we form it as cDNA for that. We use the restriction enzyme on both the plasmid and on the human growth hormone gene, and they naturally anneal. Of course, sometimes this will re-anneal with itself, sometimes this will re-anneal with itself. So we're looking specifically for recombinant plasmids. And of course, you got to have a ligase to heal the ends. So what we do is we introduce our plasmid into our host cell, in this case it's a bacterium, and then we inoculate it onto media that has the antibiotic. So if they have the plasmid, they have the antibiotic resistant gene. So we know that any bacteria containing the plasmid with the HGH gene will survive because they have the resistance gene. Boy, does that kind of sound like what we did in 201? It's just using different stuff. Gene libraries get used a lot in genetic research. The example from the book is if you were researching interleukins in humans, and there are 24 different kinds. It's really hard to study interleukins in humans. Very difficult. You don't get very many people that want to come in all the time to donate their genetic material. So what we do is we create a gene library. So what we do is we take the genome that we're interested in this figure, it would be the genome of a bacterium, but let's go back to the interleukin example. So all the interleukins, we'll say, are on one chromosome. So we take that chromosome and we chop it up using restriction enzymes, and we insert them into a vector. So we've got different pieces of the genome we're interested in, all in different vectors. We can have pieces of a gene, we can have more than one gene, or we can get lucky and we can have just the one gene that we're looking at. It's a completely random process. So what we do is we introduce each of the different vectors into different cells and we make clones of them. Now that we've got a whole bunch of copies of all of these different genes, we can pull, say, clone number 1 and take a look at what that fragment is. We can take clone number 2 and take a look at what that fragment is. And it's a whole lot easier. That's it for this topic. Here's a reminder of what I want you to learn from it, and make sure you come with questions.